The innate immune system is one of the most ancient defense mechanisms, and nearly every cell in the body is capable of mounting an innate immune response to foreign DNA or RNA. Because Phosphorothioate antisense oligonucleotides, or PS-ASOs, are nucleic acids, they can be recognized as foreign. If they're not properly designed, they can trigger this innate immune activation. Decades of work in understanding these mechanisms have already benefited patients with nano-rare conditions. We can now identify and remove about 80% of pro-inflammatory ASOs before they reach in vivo tolerability testing. But ASO technology continues to evolve, and evolve very rapidly, bringing new opportunities to design even better, more versatile drugs. A key question we're now asking is, how are PS-ASO induced innate immune responses turned off? In other words, how do cells terminate this innate immune activation once it starts? And can we control that process? Understanding this termination phase is critical. It could help us rescue ASOs that would otherwise be lost due to excessive immune activation, especially in tissues like the lung, which mount strong innate immune responses. Any advance that helps mitigate or control innate immune activation will allow us to make safer, more effective ASO therapies for patients, and to do more with our current resources. One pathway of particular interest is the oxidation of tryptophan through the kynurenine and indole oxidative pathways. This metabolic shift appears to be a key mechanism of innate immune termination, with the bioactive metabolites of these pathways representing potential anti-immunogenic co-therapies. We therefore investigated whether PS-ASO stimulation of the innate immune response engaged these pathways, and how this response is eventually shut down. What we found was that the kinetics and intensity of PS-ASO-induced innate immune responses vary between cell systems and between ASOs. We also found that the tryptophan oxidation enzymes, including IDO1 and alpha-1, are upregulated by PS-ASO-mediated immune activation. We also found that the timing and scale of these inductions differ depending on the cell system used, and that silencing IDO1 amplifies immune activation. For the first time, we've shown that innate immune termination following PS-ASO stimulation depends on a metabolic shift in tryptophan oxidation, producing small oxidative metabolites that suppress immune gene expression. These small molecules could, in principle, be included in ASO formulations to reduce or prevent innate immune activation, potentially enabling delivery to tissues like the lung, reducing side effects, and improving development efficiency by filtering out problematic ASOs early. When a PS-ASO is well designed, it enters the cell and reaches its target RNA without activating toll-like receptor 9, or TLR9, the receptor that senses foreign nucleic acids. In this case, the ASO can modulate its intended RNA target, correct gene expression, and restore normal protein levels, ultimately rescuing the cellular phenotype and improving therapeutic outcomes for patients. But when a PS-ASO is not designed optimally, it can engage TLR9 and trigger an innate immune response. We measure this activation by looking at CCL22, a downstream chemokine whose induction reflects TLR9-mediated signaling in our cell systems. This allows us to screen out pro-inflammatory ASOs early in development. Once TLR9 is activated, the cell releases a broad array of pro-inflammatory cytokines and chemokines, including IL-6, TNF-alpha, and IL-1-beta, which amplify immune signaling to neighboring cells. In parallel, immune regulatory cytokines, such as IL-10, may also be induced as part of the feedback response. The overall result is a strong pro-inflammatory cascade that can interfere with ASO function and tolerability. From this study, we observed that the degree of TLR9 activation closely correlates with the induction of two enzymes, IDO1 and alpha-1. These enzymes are both involved in oxidative tryptophan metabolism, but in complementary compartments. IDO1 functions inside of the cell, converting tryptophan to kynurenine, while alpha-1 acts outside of the cell, oxidizing tryptophan to indole-3 pyruvic acid, which can further be converted to kynurenic acid. Together, these oxidative pathways generate immunoregulatory metabolites that act on receptors such as the aryl hydrocarbon receptor to dampen inflammatory gene expression. In other words, activation of IDO1 and alpha-1 represents the cell's own mechanism to terminate PS-ASO-induced innate immune signaling. One of the first findings of our study was that both the degree and kinetics of PS-ASO innate immune responses were variable depending on cell systems and ASOs used. On this slide, we're comparing two different cell systems, each treated with a panel of PS-ASOs that varied in their level of immunogenicity. The graph on the left shows the response in BJAB cells, while the graph on the right shows the response in THP1 TLR9 reporter cells. These two cell lines represent distinct branches of the innate immune system. BJAB cells are B-cell derived, belonging to the lymphoid lineage, which give rise to adaptive immune cells, such as B and T cells. In contrast, THP1 cells are monocytic, and part of the myeloid lineage. These cells give rise to innate immune cells like macrophages and dendritic cells that act as the first line of defense. The black, dark blue, and dark red bars represent strong pro-inflammatory ASOs, while the light blue, pink, and light red bars represent weakly immunogenic ASOs. ASO 38 serves as a non-inflammatory control. Each cell line was exposed to these ASOs for 1, 2, 3, or 4 hours, allowing us to capture the kinetics of immune activation. What we observe is that BJAB cells from the lymphoid lineage show greater sensitivity and discrimination, clearly separating strong, weak, and non-inflammatory ASOs. Meanwhile, THP1 cells from the myeloid lineage respond primarily to the strongest inflammatory ASOs, with weaker responses to mild or non-inflammatory sequences. This difference reflects their biology. Lymphoid cells are tuned for selective and nuanced DNA sensing, whilst myeloid cells are programmed for rapid, high-intensity innate immune activation. Together, this comparison shows how cell lineage and receptor context strongly influence how PS-ASOs are recognized by the immune system. Building on the previous experiment, we next exposed both cell lines to the same panel of ASOs for 2, 4, 8, and 24 hours to assess how the innate immune response evolves over time. The overall trend remained consistent with our earlier results. In the BJAB cells on the left, we again observed clear discrimination between strong, weak, and non-inflammatory ASOs. However, by 8 to 24 hours, the response begins to plateau, suggesting a ceiling effect where innate signaling reaches its maximal output and can no longer increase with continued exposure. In contrast, the THP1 TLR9 cells continued to respond only to the strongly immunogenic ASOs, with little to no activation from weak or non-inflammatory sequences. This reinforces the idea that the BJAB cells from the lymphoid lineage provide a more sensitive and graded readout of PS-ASO immunogenicity, whereas THP1 TLR9 cells from the myeloid lineage display a more binary all-or-nothing response, characteristic of innate immune activation. Next, we wanted to understand how changing the concentration of PS-ASOs influenced the innate immune response in these two cell systems. To test this, we treated both BJAB and THP1 TLR9 cells with a 6-point dose range, starting at 200 micromolars and decreasing in intervals down to 40, 8, 1.6, 0.32, and 0.064 micromolar. This approach allowed us to determine whether dose escalation amplifies or alters the immunogenicity profile of each ASO. In BJAB cells, we observed the expected bell-shaped dose-response curve. Immune activation increased with dose up to around 1.6 micromolar, after which it declined, likely due to toxic effects at higher concentrations. Interestingly, ASO 18, one of our weakly inflammatory sequences, showed a steady increase in immune activation across all doses, without the decline seen in other ASOs. In THP1 TLR9 cells, we saw a similar overall pattern, but the signal intensity was substantially lower, consistent with the reduced sensitivity of this myeloid system compared to the more responsive lymphoid BJAB cells. Together, these results suggest that dose strongly influences innate immune readouts, and that BJAB cells provide a more dynamic and sensitive system for assessing PS-ASO immunogenicity across concentrations. From the previous experiments, we've seen that innate immune activation by PS-ASOs is cell type specific, time dependent, and dose dependent. Taken together, these findings suggest that the magnitude and kinetics of innate immune activation are tightly regulated and may involve built-in feedback mechanisms within the cell. To explore this further, we turned our focus to tryptophan metabolism, and specifically to the two enzymes that control its oxidation, IDO1 and alpha-I1, to understand how these pathways respond to different PS-ASOs and whether they contribute towards terminating the innate immune response. To investigate this relationship, we measured TLR9 activation across our panel of PS-ASOs and performed a correlation analysis with the expression levels of IDO1 and alpha-I1. Interestingly, in both cell systems, we observe a significant positive correlation between the degree of TLR9 activation and the upregulation of these two enzymes. We observed the strength of this relationship increase when ASOs were further segregated into rapid and slow TLR9 agonists, shown by the blue and red dots. As expected, slow TLR9 agonists produced lower levels of alpha-1, IDO1, and CCL22 compared to fast agonists, even when only considering the peak response values for each experiment, as shown here in Figure B on the right. These results suggest that tryptophan oxidation enzymes are tightly coupled to the intensity and kinetics of TLR9 activation, linking innate immune stimulation directly to the cell's metabolic feedback response. Building on our earlier experiments, we next examined the kinetics of messenger RNA expression for IDO1, alpha-1, and CCL22 in both the BJAB and THP1 TLR9 cell systems. To do this, we measured gene expression at multiple time points following ASO treatment. This included 8, 24, 48, and 72 hours. This allowed us to capture both early and sustained responses. Consistent with our previous data, we saw a strong temporal coupling between CCL22 induction and IDO1 or alpha-1 upregulation in cells treated with the strong immunogenic ASOs, ASO CpG, 95, and 20. When CCL22 expression increased, so did IDO1 and alpha-1. When CCL22 declined, the oxidative enzymes followed the same trend. In contrast, the non-inflammatory ASO 38 produced only minimal induction of IDO1 and alpha-1, confirming that well-tolerated PS-ASOs do not trigger TLR9 activation or engage the compensatory kynurenine pathway. Once again, ASO 05, 12, and 12 upregulated CCL22 and alpha-1 in the BJABs, but had little to no effect in the THP1 TLR9 cell system, reinforcing that these ASOs are non-immunogenic in this cell system. Overall, we found a proportional and time-dependent relationship between TLR9 activation and IDO1 or alpha-1 expression across both models, indicating that tryptophan oxidation serves as a downstream regulatory mechanism for terminating PS-ASO induced innate immune signaling. So far, we've seen that IDO1 and alpha-1 expression track closely with TLR9 activation over time, suggesting that tryptophan oxidation might serve as part of the cell's feedback response. To test whether this transcriptional upregulation translates into functional enzyme activity, we next measured tryptophan consumption and kynurenine production following PS-ASO treatment. To confirm that the increase in IDO1 messenger RNA we observed actually reflects functional enzyme activity, we next measured the consumption of tryptophan and production of kynurenine following PS-ASO treatment. We performed a dose-response study at 0.32, 1.6, and 8 micromolar, and collected samples at 24 and 48 hours to assess both early and peak activity. While all PS-ASOs caused some decrease in tryptophan, only those that produced a simultaneous increase in kynurenine were considered to have active IDO1 enzyme. In the THP1 TLR9 cells, we observed a significant increase in kynurenine coupled with a significant decrease in tryptophan after treatment with the ASOs CpG, 95, 20, and 18. Across all PS-ASOs tested, peak IDO1 activity occurred at 48 hours, and the activity scaled with dose, consistent with the dose-dependent induction of tryptophan oxidation. To further explore how this metabolic response feeds back into the immune pathway, we next asked whether silencing the tryptophan oxidation enzymes, IDO1, would alter the magnitude of TLR9 activation itself. We next wanted to test whether tryptophan oxidation itself is directly responsible for suppressing innate immune activation. To do this, we asked a very simple question. What happens if we reduce IDO1, the enzyme responsible for roughly 90% of the tryptophan conversion to kynurenine? We treated THP1 TLR9 cells with siRNAs targeting IDO1, which resulted in about a 90% knockdown of IDO1 protein, as shown in the immunoblot on the right. After reducing IDO1 levels, we re-stimulated the cells with a CpG ASO and found that TLR9 activation nearly doubled compared to untreated cells. This indicates that IDO1 acts as a critical negative regulator of innate immune signaling, helping to dampen TLR9-driven activation once the response has begun. And interestingly, this suppression may be partially maintained by alpha-1, particularly in cells that express both oxidative enzymes. Taken together, our findings identify and characterize the immune signatures of PS-ASO induced TLR9 activation across two distinct cell systems. We show that the tryptophan catabolizing enzymes IDO1 and alpha-1 play a central role in generating kynurenine pathway metabolites that help quench the innate immune signaling. These results open new opportunities to explore kynurenine pathway modulation as a strategy to mitigate PS-ASO induced innate immune activation. In the longer term, it may even be possible to incorporate kynurenine pathway metabolites directly into ASO formulations, or to use them as co-therapies to improve tolerability, though this will require further mechanistic and translational studies. While our work provides clear mechanistic insight, we also recognize that the immune environment in vivo is far more complex than what can be fully captured by two cell lines. Still, this study provides compelling evidence that the kynurenine pathway functions as an intrinsic regulatory axis for PS-ASO-mediated innate immune signaling and establishes a strong foundation for further research into its therapeutic potential. In summary, our results highlight that the kynurenine pathway metabolites may serve as a tolerogenic co-therapy for antisense oligonucleotides, offering a path forward towards safer and more effective RNA-targeted medicines.